Hi guys, so the sheet that Ms. K just went through with amoebas for you guys for your labs for notes or your notes for lab. Can you guys take that page, flip it over? On the other side, you guys are going to see the paramecium, which is the second organism you guys are going to watch a video on. On the back side of that particular sheet, you will see everything that you need to know for your paramecium. So paramecium is a little bit different than amoebas in terms of shape. So it's more of an oval slipper-shaped molecule rather than a blobby shaped structure like you saw with the amoebas. It does have a rounded front top and a pointed back or bottom end of it when you guys look at the diagram and see it on the video. A couple parts that are a little bit different than what we have with our amoebas. So it's got more of a pellicle rather than a cell membrane or plasma membrane. It's a little bit more stiff, but it's still elastic membrane. It's going to give it more of a definite shape, so it's not constantly changing shape like the amoeba is. Covering the pellicle, you guys will see on the video little tiny hairs called cilia. The cilia are going to be used for movement. It also has a contractile vacuole, just like your amoeba did. It has the same exact function as your amoeba, and that's in order to pump out that particular water. Remember, if they're not able to pump out water since they live in a water environment, they will go through and explode, kind of like putting too much air into a balloon and that balloon popping. We need a way to get it out. So that's what the contractile vacuole does. Since we said they have kind of this flat top, rounded area on the top and more of a pointed back or bottom end, they have an anterior and posterior end. The anterior end is near the head or the front portion of them, and that's the end that's going to be a little bit more rounded. The posterior end is near the rear, and that's going to be more of the pointed or back or bottom end of them. When these guys go through and eat, what you guys are going to see is that they actually eat these little microorganisms, so tiny little organisms like bacteria or algae or yeast, things that you guys may have seen underneath the scope when we did our microcosms. They use their cilia, those little hair-like structures that are going to sweep the food along with some water, and they move it into kind of what is like a little mouth for them. And then it forms a food vacuole at the very end. When the vacuole reaches what is known as an anal pore, meaning that vacuole moves throughout the organism, they digest down the food, that little vacuole with the waste material is going to hook up to what is known as an anal pore, which is like a little opening that they have, and then the undigested waste can be removed from it. Just like with your amoeba, anything that's being released from that particular organism is exocytosis, meaning it's exiting, it's leaving that particular organism. In this case, it's going to be waste material. In terms of locomotion, paramecium swims by beating their cilia, so they're moving those short little hairs back and forth, which is going to allow them to move around in that pond environment that you'll see. Underneath all those notes, you guys will see a little diagram that looks like this. Once again, we'll talk about it. You'll see another diagram within your lab sheet. So you guys want to kind of organize yourself, and at this point, kind of pull out your lab sheet that looks like this, and we'll be going through this in the next video.